Good morning, everybody. Welcome to the Informatics Power Talk Friday seminar series. I'm very happy to introduce today's speaker. It's our very own Dr. Stephen Barnes, Distinguished Professor of Medicine in the Division of Pulmonary Allergy and Critical Care Medicine, and Director Emeritus of the Targeted Metabolomics and Proteomics Laboratory at the School of Medicine at UAB. He has a very distinguished career, originally chemistry and biochemistry in the UK, and after joining UAB has studied many areas of proteomics, bile acids, isoflavones, and has many distinguished awards. And kind of like we had a talk last week about the applications and of metabolomics, and this is sort of like the other part of that is talking about the data part of it. So without further ado, Dr. Barnes.
Well, thank you very much, Amy, and everybody else who is online. It is Good Friday, and I appreciate you being here today. So as Amy said, what I'm going to concentrate a lot on today is about the data that underlines metabolomics. So let's start by looking at what I'm going to try and cover. So where did metabolomics come from? And I have a personal part to this. The metabolomics at NIH and UAB's contribution to it, and then the metabolomics data and data interpretation, and an area that I think is very controversial, the ethics of doing metabolomics in clinical studies. And then finally, we'll talk about an iron feature, a nature of the data in LCMS, and what it leads us to is discovery, something that we just didn't know about before and which has profound implications. So let's get going. As a very young investigator, I had worked in the lab of Tony James, who was the inventor of gas chromatography, as an undergraduate, and suddenly found my passion for what I wanted to do, which was to become a Ph.D. and move into an academic career. And I joined the lab of Sir Ernst Boris Chain. Chain was the 1945 Nobel laureate, along with Fleming and Florey. He was the biochemist/chemist who chemically characterized penicillin and allowed penicillin to become what it was today, one of the first antibiotic that we could take. And he was also, if you read his Nobel Prize summary, you also realize that he was renowned for his work on microanalysis. So what he was doing in the period between 1945 and when I joined his lab was to study the metabolism of major compounds, things like glucose. And so he was giving cells and hearts and livers carbon-14 labeled glucose, radioactively labeled glucose, and then using paper chromatography in 2 dimensions to resolve the intermediates of glycolysis, the Krebs cycle, and amino acids. And once the paper had the two chromatographies on the paper had been done, then they were mounted onto this frame here, and a Geiger counter was brought up close to the surface of the paper, and for each letter, as it were, of the typewriter that it was mounted on, it recorded the radioactivity and then moved over one letter and then the next letter and so on to the end of the line, and then stepped down a line, just like an old typewriter, for those who can remember old typewriters. Anyway, it allowed then at each spot in that 2-dimensional space to the data, or the signal there, to be digitized. And therefore, with the data output, then you could put that into a computer and then generate figures and other types of interpretations. And so this was metabolomics. It wasn't called metabolomics then, but in essence it was. So it was chromatography to separate compounds, it was digitizing the data, and it was using computer programs. And he did this alongside another guy who was very influential for me at the time, Keith Mansford. And Keith was the chief scientist at Beecham's Pharmaceuticals. And this is me. What I brought to the group was the ability to do gas chromatography to separate those same compounds, glycolytic intermediates and Krebs cycle intermediates, but I did it in a GC. This is the GC oven in here. And when the samples went through the columns and came out, part of the sample went into a flame ionization detector, and the other part went down a heated tube into this apparatus, which measured the radioactivity. These are two photomultipliers that were looking at a cell that's in there in the center. And so all of that was digitized. Here's the digital tape generator. And we could record 1 data point per second. Such was the mechanical nature of forming the paper tape recording. Initially, I had to take that tape over to a terminal where I could have the tape read by a computer over in University College. But towards the end of my time there, we got our own computer, which was about the size of a large fridge and had a memory of, I think, 8K. But it enables now to, in real time, take a look at the chromatograms. So we were able to get a visual image of what we were doing. And I applied this to the acellular slime mold, Physarum polycephalum, and the group at Imperial applied it also to the beating heart preparations. I also went on, when I got to UAB, to take advantage of the superconducting NMR capability that UAB acquired in the early 1980s and applied this to sort out the proton spectrum of bile acids. I then worked with Gabi Elgavish, who came here to UAB, and he taught me how to use rare earth metals like dysprosium and other rare earths to sort out the structure of bile acids in solution. So I had this a little bit of an NMR experience, a lot of chromatography experience, and I also have become the mass spec person here at UAB. So all of that led to the NIH deciding that I was the right person to organize a workshop on metabolomics. Metabolomics began as an investment at NIH in 2012. There's the result of investment through the Common Fund. I have to say I was on the Common Fund Council of Councils committee, and I actually voted for metabolomics prior to this. Anyway, because of that, and also because of Hemant Tiwari, who had been organizing training workshops in other disciplines here at UAB, we were able to put in an effective proposal. And so this is the group of people who were trained here in, this is the 2015 workshop. We had 6 of these. Altogether, we trained 200 people, mostly graduate students and postdocs, but some early emerging investigators, a couple of core facility directors from outside UAB, and one NIH director who came down and took the course. Because of this, putting all this together, this allowed me, because I built all sorts of different components as part of the workshop, enabled me to construct a graduate course on special topics in metabolomics. And we started that in 2015, and I teach it every year, and I've been doing so ever since. We literally just finished this year's course. In 2016, the bioinformatics team at NAID requested that could I give this course again to students at a university in Mali, the University of Sciences, Techniques, Technologies of Bamako, and do this for the African Centers of Excellence program. And in fact, tomorrow I'm giving a webinar on metabolomics to Metabahol, a company that was formed by Ola Kunle Jai Yasimi who's a postdoc at Harvard for students, fellows, and investigators from Africa. And it turns out from many other countries from learning about who signed up. I will be this year in the fall teaching two workshops. One, a very simple one-day workshop on how to process metabolomics data, and then a longer 5-day one, which will dig into the different procedures that are available for interpreting metabolomics data. Okay. So what are metabolomics data? I'm really going to talk about LCMS and not about NMR. For LCMS, they are a separation number. That's something that is in time units. You know, in the 2D paper chromatography, it was distance, but for us, for LCMS, we're passing compounds through a column, and we note the time at which they emerge from the column. So that's one of their characteristics. And the other characteristic is their weight or mass. So what is mass? Metabolites will form ions. So that's what we're going to measure. But all metabolites, and in fact any other chemical, have an empirical formula made up of a combination of elements that are joined spatially. So here's an example. This is phenylalanine. We have a phenyl ring over here, and then this is the part that all amino acids have. So this is the side chain to the amino acid. So that's phenylalanine. The mass of each element has been determined absolutely and is measured by others. If you go to NIST, you can get the measurement for an individual element to at least 9 places of decimals. Because of this, very simply, we can predict the mass of a metabolite by adding up the number of atoms of individual elements. So typically carbon, hydrogen, oxygen, nitrogen, and maybe sulfur, and maybe phosphorus. Those are the ones that we find in biological samples. And once we know the mass of the metabolite, then by adding a proton or subtracting a proton, we can get to the molecular mass form that we will see in when we do metabolomics. Very interestingly, and this is we use this a lot, when you standardize every mass to carbon-12, the major isotope of carbon, it's realized that they have non-integer mass defects. In other words, they're not integers. And the physicists latched onto this in part to create nuclear weapons, which we didn't want, but also defining for us why our sun works, and in fact the reason for the existence of our universe and us. So it's really, really fundamental to know about the mass of an element and the mass of a compound. So how do we go about acquiring LCMS metabolomics data? We put our sample onto a column. Some samples come off some metabolites come off very quickly, depending on how they interact with the column. Our columns are what we call reverse phase. So hydrophilic compounds, water-soluble compounds, come off very quickly. And the ones that are more hydrophobic, rather like the phenylalanine, which has that phenyl group on it, come off more slowly. And we cause the elution of the more hydrophobic compounds by increasing the level of acetonitrile over a gradient. When we're done, we need to be able to measure the area under a peak for a given compound. And we've shown that, and others have shown that you need at least 10 data points to do this accurately. So since the peak is 3 seconds wide, we chose to have a duty cycle of 4 Hertz. In other words, we're collecting a series of pieces of information every 250 milliseconds. So we start in the first 100 milliseconds of the duty cycle, collecting a picture of everything. So from masses from 50 up to 1,000. So this detects all the molecular ions, so the negatively charged M minus H ions and the positively charged M plus H ions. The remainder of that 250 milliseconds is a time where we collect MS data or mass fingerprint data. And so one at a time, 30 molecular ions are selected. They're filtered by the quadrupole, accelerated, and then crashed into a puff of gas. And this heats them up and breaks them into pieces. And these pieces are very reproducible. So we do that for about 5 milliseconds, and we use the TOF analyzer to measure them. As you might imagine, this generates a lot of data. So if we're running something that is 6 to 7 minutes long, that's, let's say, for our sake of argument, 400 seconds, and we're doing this 4 times a second. So we're collecting 1600 spectra, but in terms of the MS spectra, it's many, many times more than that. So the data sets are very big. So this is what a total ion chromatogram looks like. So this is all the ions that were eluting from the column over the period of the experiment. So we have at this low end here, we have a low, if zero level of acetonitrile. And as we move across here, we get to the maximum level of acetonitrile. And then there's a wash period here. So as you can see, most of the ions are coming off by 6 or maybe just after 6 minutes. If you look very, very carefully, you might see 3 colors. This is because this sample was run 3 times. So you can see that it's pretty reproducible. Now remember the phenylalanine. It has this empirical formula, C9H11NO2. And from that, we can deduce what its molecular weight is. It's going to be 165 plus. And of course, the negative ion of it will be approximately 1 Dalton less, so it will be 164.072. So we can filter the data we got above, and now we see the phenylalanine in all its glory. Okay, so this is an example, phenylalanine, of an ion feature. It has a retention time of 1.72 minutes and an M/Z mass of 164. And this will be reproducible sample to sample. The size of the peak may change, and that's what we measure and then begin to interpret. So how do we do it? I mean, if you had to manually interpret these 1600 spectra plus all the MS spectra, there's no way that you could do this. So, and unfortunately, well, not fortunately, but it's a practical issue. The manufacturer records the data in a special format, and they do that so that people can't tamper with the data and alter it. And that's very important for the pharmaceutical industry. But they work with two of the options that one has for doing data interpretation. XCMS, which is an online program as well as being an R program, they have provided an API to convert each individual manufacturer's raw data into a format that's useful and can be used by XCMS. And the same for MS Style. We started using XCMS actually more than a decade ago. It's now become commercial, which often happens with freeware. Eventually, they have to be supported by somewhere. So in 2015, we shifted to using MS Style. And the very nice thing about MS Style is it has a GUI. You can only do it on a PC. You can run the program in other platforms, but you don't have a picture. And the nice thing, as you'll see in a bit, is that MS Style gives us an integrated visual analysis. There are other programs, and they have to undergo and need the requirement of converting the raw data. And there's a program that's a freeware that's called MS Convert. It's part of ProteoWizard, and it allows these 3 other programs to work. MC Mine, which is a community software, works on a PC and a Mac. ADAP Big, which was developed by Shusha Du at University of North Carolina in Charlotte. That also runs on a PC Mac. And then quite recently, Asari has come along, and it's really very interesting program. When I had a study that I was running on MS Style that was 824 different samples, and I used my laptop, which had 32 megs, 32 gigs of internal memory and a terabyte of SSD. It took 2.5 days to run. When I ran it on Asari, it took 10 minutes. So it still needs to be perfected, but that is a really interesting development in the field. So each one of these programs then produces an output. It gives the lists of different ion features, and where it's possible, they are annotated. And that depends a lot on the databases that can be used for comparison between the observed MS-MS spectra and the ones that are in that database. Once that data is cleaned up, then, excuse me, went the wrong way, you can submit it to a very, very good program called MetaboAnalyst. It allows normalization of the data in multiple ways. It will carry out univariate and multivariate statistical analysis, heat maps, random forest machine learning, et cetera, et cetera. It's now, there is a little fee if you want to get organized outputs, but you can still download all the data you capture doing MetaboAnalyst. And so this is what MS Style looks like. And so in this particular example, I think this is from a lady at the University of Wisconsin that we were working with. This is an example of clicking on the data to identify and get information about an individual metabolite. And this is phenylacetylglutamine. It's actually uremic toxin. So if it goes up in your urine, this is a bad sign. So if you go over into this box here, you see the MS-MS spectrum. And hold on, let's go back one, sorry. The MS-MS spectrum. So the upper one here is the observed MS-MS spectrum that we got for this compound in the sample. And the lower one is what is in the public databases for phenylacetylglutamine. And we see several ions in perfect conformation between these two. Interestingly, besides seeing the M-H or the 2 or the M plus H, if you're working in positive, you see other adducts that are being formed in the gas phase as compounds go from the liquid into the gas phase. That a Nobel Prize was given for the person who invented that electrospray technique, John Fenn, and it was done for proteins, but in fact works really well for almost any compound. But anyway, so you, among all the different features that you see, not only do you see the M-H or the M plus H, the molecular ions, but you also see other adducts that are formed. And so one of them here is just simply, if I can get it, here we go. This is simply the dimer of the compound because it was in high amounts. So this, over here, we see what was in the all the patients, the mean and standard deviation of that. This is a pooled sample run multiple times during the run, and it should be approximately the average of this one. And then this is a sample that was used to do MS-MS work, and it's reproducing extremely well. And different amounts of it were injected compared across these samples. And then over here, these are just the blanks we run. So we want to make sure that when we're measuring something, it's not coming through the procedure from non-biological sources. Okay, so this is another example from these patients. And you can see that there was quite a lot of people had this. This is now a drug. It's actually a drug metabolite. It's the glucuronide of Tylenol. and it also forms dimers and possibly trimers. Its MSMS spectrum matches very well between the observed and the what was in the public databases. So we like this about MSStar because it visually allows you to look at all the data. Okay, so in that case, we were looking at some samples that were coming from clinical state, but we do many other studies in which we have samples from clinical circumstance. And this can be very helpful. It can assure that the patient is taking the drug that has been prescribed for them. But we also see everything else they're doing, and sometimes perhaps we get to see too much. That was something that we discussed at the end of Susan Sumner's meeting last week. So there are two ways that we can go about analyzing the collected LCMS data. We could match the MSMS spectra against a metabolite intermediates database. When we do that, we will not see the drugs that the patient is taking. On the other hand, if the drugs that the patient is taking is influencing the result of the primary experiment, that may not be what we want, and we need to see the whole story. So the alternative is to run against a database of all molecules that were currently in common fund or in publicly available databases. And so we did this in a group of patients who were recovering from spinal cord injury and were placed on a regular diet versus a ketogenic diet. There were 36 patients who took part in this study. All of them gave a baseline sample, but only 18 returned to give the second sample. When we looked at the data, we made a pooled sample of everybody, and that was actually of all 54 patients, not the 18 that came, or the 54 samples. The 18 patients who provided a starting sample and then came back and provided a second sample after going on these different diets. These were analyzed separately, but the pooled sample was all 54 samples. And I was checking out, trying to make sure that what I saw in the pooled sample was an average of what was in those 18 samples or 18 patients and the two samples they give. And I found a discordancy. So this one is actually a picture of trazodone, which is one of the drugs that they were taking. Let me point out this first, that the mass spec sees everything regardless of what the chemical is in the sample. And so you may have noticed this rising series of ions here, and I think there's a second one there. And these turn out to be polyethylene glycols, which are coming both from how the drugs are dispensed and given to the patient, and also coming from plasticware that the sample was put in before it reached our lab. So trazodone gives an MSMS spectrum that matches very nicely. And so we were able to determine which patient was taking the trazodone. But we had this problem compound that was high in the pooled sample, but we couldn't see it in the patients. And let me go to the next one. And it turned out to be cocaine. And so the reason why we didn't see it in the 18 who came back for the second sample was that they basically weren't being exposed to cocaine. And you can see the MSMS spectrum confirming. So this is cocaine. And so we went back to the 18 patient samples who didn't return and analyzed those. And we found that 12 of those 18 patients were the ones who were either taking cocaine or had cocaine metabolites in their blood. So I guess we learn from that the patients who don't come back might be doing so for a reason. In this case, we found out what the reason might be. But anyway, when we went back, looked at all the 54 patient samples, we found a total of 84 drugs, both therapeutic and over the counter, and each one of these was verified by MSMS. So then we asked the people we were working with, what medications are they taking? What's in the clinical record? And only 14 of that 28 were actually drugs. The rest were infusions like amino acid infusions, electrolytes that we used to support this particular group of patients. When we took those 14, we said, okay, can we now assign those drugs correctly to each of the patients in the study? And we were able to do so. So metabolomics can provide a very careful description of what is in the individual patient. So it raises some ethical issues, particularly if the consent form, did not say that we will see everything that you're taking, and if they didn't say that, then obviously this information can't be used. So how can we do LCMS metabolomics or measurements a little bit more ethically? So we've been working with Dr. Siddiqui here at UAB, and he approached us actually quite a few years ago to ask the question about, can we develop assays to be used in studies of compliance for the use of antihypertensives? And apparently there is people who take antihypertensives have a very bad record of compliance. So an LCMS assay that is now targeted to the antihypertensives, in other words, not trying to measure everything, but just saying let's measure only the ones that you put in a list, then that would be much simpler, a bit rapid, as you'll see, and perhaps and definitely more ethical. So Dr. Siddiqui is in cardiovascular medicine here at UAB, and he works with his colleague in England at the University of Leicester, Pankaj Gupta, and his his grad student has developed an assay for these large number of antihypertensives that can actually occur in 45 seconds per sample, so really, really fast. And as a consequence of this, we're assembling an application to the UAB HSF program to bring his student over here and to develop this assay. In England, there is a National Centre for Adherence Testing that supports the use of clinical drugs across many of the National Health Service hypertension centres. So at this point, I thought I'd stop here. We've got about another 10, maybe 12 minutes of, I wonder if anyone has a comment here. Please intervene, because obviously we talk about personalized medicine here at UAB and the electronic health record. And so is metabolomics helpful, and how could it be helpful? I can't see anybody, so is there anyone who's interested to comment? You can always send me your comments by email. I'd appreciate that, because it is an important issue to carry out personalized medicine in a really effective way. Okay, well, no matter. I wanted to finish with something that's come up that's really, really, really interesting. And so let's first of all go back to what the LCMS is seeing. So remember, we're talking about iron features, and this is a multi-level assurance issue. So it's based on retention time, particularly if you've built a database that's exactly the same as the one that you're using for analyzing samples. It definitely depends on the mass of the molecular ion, which we can measure very accurately to 1 or 2 millidaltons or even better. And also the MS product ion fingerprint. So that's how we annotate. So it depends, as I said, it depends on in-house and public databases. What we find is that we can collect this very large amount of data, but the ions that we can truly annotate with confidence is actually only a small percentage of the total. Sometimes it's as low as 2%. Even with the best way in which people do it, maybe we're no more than 10%. So there's an awful lot of data there. Some of it is adducts. You saw some pictures of the adduct formation in association. So an adduct will come off at the same time as the compound, because the elution time is dependent on the mobility of the compound before it becomes an ion. So any of the adducts it's associated with will come with it. And then there are natural isotopic forms of carbon, particularly. We're all 1.1% carbon-13. So other compounds there. Well, let me now sort of shunt you into the world of bile acids, which Amy mentioned is one of my strong areas of interest. And so these are the most abundant species in the GI tract. They're made in the liver from cholesterol, and then they're conjugated either with glycine or taurine in the human. So this is the cholesterol molecule, and it's a sterol, and it's converted through a multitude of steps to remove 3 carbon atoms in the side chain and to oxidize the side chain so it becomes a carboxylic acid. And it also adds hydroxyl groups into the rings and gets rid of the double bond that you see over here. So these compounds become very good detergents, and in the upper part of the small intestine, they are, in this form, they would be insoluble. So they're conjugated either with glycine, that's shown here, or with taurine, which is shown here, these 2 amino acids. So they're crucial because of the detergent action, they're crucial for dispersing fat, aiding in lipolysis, and then transmitting through micelles the results of that digestion lipolysis to the intestinal wall, and hence the uptake. They also facilitate the uptake of drugs. And with Charles Fellaini and with Martin Johnson, we were able to isolate, first of all, the enzyme from human liver and make antibodies to it, and then use the antibodies to recover the enzyme. We call it BAT. So BAT was cloned from a cDNA library, expressed in humans, and then we also did that in mice and rats. Interestingly, mice can only make taurine conjugates, and that's actually true for almost all species, except for mostly the higher apes and 1 or 2 other species that are capable of doing glycine conjugation. Now, in the test tube, BAT only makes taurine conjugates. It will not make any glycine conjugates. But if you look in mouse bile, then nonetheless there is a small amount, about 1%, of glycine conjugated bile acids. So I was actually presenting a poster about 6 years ago at a meeting in Atlanta, and about the bile acids in feces of people from here in Alabama. I was seeing the glycine and taurine conjugates, particularly the taurine conjugates, in large quantities. On the other side of the poster was Robert Quinn, who was trained or was a postdoc in Peter Dorostein's lab at UCSD. And he did one of those fundamental science experiments that NIH studies section probably would not fund. So he was curious, what happens when you compare the metabolites between a wild type mouse and a germ-free mouse of the same strain? So he took, I think it was 4 animals, divided the intestinal tract from nose to anus into small pieces, and then performed LCMS on each one. He had no interest in bile acids at this point. As I said, he was really looking for metabolites that were unique to animals that had bacteria. When he did this, most of the, as I indicated before, most of the compounds, and particularly the bacterially produced ones, were represented unknowns. And so he subjected that data to network analysis through software that's available online called GMPS. So let's take a look just briefly at the a mass fingerprint. This is the product ion spectra of glycocholate, so a bile acid that everyone on this call has lots and lots of. And what you see in the positive ion MSMS spectrum are several things. The first thing is that when you get rid of the side chain and the hydroxyl groups off of the choline, you end up with this ion at 337.2538. That is seen in every trihydroxy bile acid. Cholic acid is a trihydroxy bile acid. So it's an ion that characterizes that type of bile acid. You also notice here that, and if the individual masses are not given, but I assure you they represent 3 water losses. And so you can see that going from 466 to 412, there's a loss of 54 to produce this very intense ion at 412. You can't see it over here, but there is a small peak that is coming from the glycine. So I was working with Dr. Youngmi Kim's study. She had given us some fecal bile acids or fecal samples to run, and we saw in this the mass spectrum this particular MSMS spectra. Notice it's got that 337 here, and it's got some what appeared to be water losses. We didn't see the other ones here, but my suspicion was that this one lost water more easily than the glycocholate did. And we have an ion at 502. So there's the trihydroxy bile acid signature. And what we also noticed, what we also noticed was this new peak at 166. So in mass spectrometry, you do a lot of simple arithmetic in your head. And so we notice here that sorry, let's go back up again. The peak at 412 for glycocholate has moved to 502, which is this unknown. And that's a mass difference of 90, plus a little bit. If we go to the glycine had a mass of 76. And this new peak is at 166. And that difference is also 90. So what is 166? I wonder if anybody on this call can figure this out. If you listen carefully to what I said, you should be able to tell me that this is phenylalanine. We saw before that the phenylalanine had a mass of 164, but that was in the negative. So in the positive, we've added 2 protons. We've added a hydrogen atom and a proton. And so the mass of this, of phenylalanine in the positive is 166. So let's look to see what Robert Quinn found. And he gave a talk here, which is, I show you the link below there that you can go and listen to the full talk. He's a wonderful speaker. So this is the network analysis that he did. And so he was looking for compounds that were unique, were green, that were unique and different from compounds that were only found in the sterile component compartment or were shared in both types of animals. And so up in this top corner, you see this little collection around, he called them the glycocholic acids. We see them in slightly more expanded view here, and again here. And so the 466 is the ion that you would see for glycocholate. This is the 556 that is the 54 plus the 502 peak that we had seen in the MSMS spectrum. And then you also see here the difference, this 572, the difference in mass was 15.995. And if you're a mass spec person, you immediately know that's oxygen. And so what he found and reported in the Nature paper was phenylalanine, and then, of course, the hydroxylated phenylalanine tyrosine. He also reported leucine and isoleucine, and these are the three structures of these compounds. So, all very interesting. But then during COVID, his lab manager was able to get some bat knockout mice from UC Davis. Dr. Fellaini and I had tried to make a knockout about 20 years ago, and we ended up with an animal with too many chromosomes, so we weren't able to use it. But anyway, he bred them to make double knockouts and studied their growth. And they grew poorly for the first 3 weeks, as we all predicted, and that included me, because we weren't going. The animal would not be able to make any bile acid amidates because we had knocked out this hepatic enzyme. But after 8 weeks, their body weights had come back and matched those of the wild type. So they took a look to see what was going on, and they went into the bile and they found it had high concentrations of bile acid amidates. The choline, which was the one that was being made by the liver, was 27 times lower than wild type mice, and suggests that, yes, the knockout had occurred. The glycine, the choline was about the same as in the wild type mice, but there were a whole slew of other novel bile acid amidates. And so what had happened in response to the knockout, that the body turned to other mechanisms to make bile acid amidates that provided the detergent-like properties that were needed to allow the animals to grow. So how does this come about? About the same time as I was running into Robert, Zhu et al. Had crystallized the bile salt hydrolase, the enzyme that removes the glycine and taurine groups from the bile acid. And they came up and showed a picture of where the glycocholate binds in the active site. The key group that's needed for this enzyme reaction is this cysteine group, cysteine 2. It's a water-exposed cavity in the molecule, and the cysteine is responsible for the cleavage of the glycine away. It leaves the cholic acid in the active site, but it also means that while water is the driver for this, and that's how the enzyme was being considered, that it was a hydrolase. In fact, water is just a weak neutrophile or nucleophile. So in principle, any nucleophile could do it. And so as the intestinal contents pass through the colon, this is a multi-day process, the water is gradually removed and many small molecules are released, including amino acids and other amine-containing compounds that have strong nucleophile centers. And so therefore you can actually get a reverse of this reaction. And I looked in the data that I got, the fecal samples from Youngmi Kim, and this is plotted in a log-log plot, and you can see that there are many different amino acid conjugates that were being formed. These are the ones that I had libraries with that I could compare the MSMS spectrum. Okay, so where does this leave us? Metabolomics and lipidomics, I haven't even started on that. That's also very important, is an interesting and quite challenging area of data science. It opens a window into the chemical world that surrounds genes and proteins and determines their expression and functionality. A big new issue is doing imaging mass spectrometry to actually locate where the metabolites are juxtaposed to genes and the gene products. In clinical medicine, metabolomics reveals everything. and all the medications a patient is taking. But it also leaves a lot of ethical issues. So maybe targeted metabolomics could prove to be more beneficial in clinical studies. It's certainly ethically more simpler. And network analysis has been pioneered by the UCSD group, is allowing understanding of what we might call the dark metabolome, the unannotated part of the metabolome. And what we also get from this is metabolomics reveal that bacteria can offset the loss of genes and their function. There's a lot more developing in this area, and it turns out the bacterial species that have this enzyme activity have quite a range of hydrolase versus amidating capabilities due to subtle amino acid sequence variations. So not every bacteria is actually going to be an amidating bacteria, even if it has the hydrolase present. And therefore, your colonic microbiota are absolutely critical as to whether you can receive benefits from processes like this. So finally, I'd like to thank a lot of people. First of all, my mentors, Tony James, who taught me about gas chromatography, my PhD mentor, Ernst Chain, Dame Sheila Schulich, who taught me about the liver and bile acids and sent me to Alan Hoffman's lab, who became my friend for life, sadly died in 2021. And then I'd like to thank Charles Fellaini, Karen Yarra, Shah Lee, Robert Youngmi Kim, and of course Robert Quinn. And then Hemant Tiwari, who enabled us to have the workshop here at UAB. And then the three people who taught me about data analysis of metabolics data, Shusha Du, Shushao Lee, and Paul Benton. And a call out to Landon Wilson, who's worked for me for 25 years now and is the person who produces all the data that we do the analysis. And then finally to NIH for my bile acid work, the workshop, and for instrumentation. Thank you.
Well, thanks for the great talk. We do have a few questions.
Yep.
And you can see them in the chat, or I can also read them out to you.
Okay, let me get out of this so I can.
In the meantime, anybody has questions, please feel free to enter them in the chat, or we can also, you can also push the raise hand button.
Okay. Database for drugs. Who maintains it? Well, if you go to the MS Style website, there is a very large database which contains many drugs. Back in 2020, when I realized that how well metabolomics could identify drugs, I went to Bruce Korf and said, Bruce, would you invest in you can get from companies all the current FDA approved drugs. They're typically used for people who are doing cell culture experiments to measure how a cell responds to a particular drug. I would need just a tiny fraction of each one of those. So I said, would somebody put money up for that? Well, sadly they didn't. But there are, I think, 2,500 drugs that you could do. And we could create a database of all the drugs and therefore go and look at studies that are being done in patients. So it could be done. So that was the database. Yeah, somebody asked about of course, to cancer research. In fact, when the National Cancer Institute really started exploring ways to prevent or to treat cancer back in the late 1940s, a major emphasis was on things that interfere with metabolism. And in fact, we've come the full circle back. So one of the drugs that, or one of the enzyme that turned out to be really crucial in different types of cancer was isocitrate dehydrogenase, or IDH. And that is a key enzyme in the Krebs cycle. And there are mutations of it that prevent it converting isocitrate to alpha-ketoglutarate. Alpha-ketoglutarate is a absolutely key enzyme in the body because it is the cofactor for something called HIF-1 alpha. So HIF-1 alpha is called the oxygen sensing protein. It's in the cytoplasm and it has a partner in the nucleus, HIF-1 beta. When the two of them get together, it turns on all the genes associated with cancer. It is the HIF-1 alpha is converted in, it's hydroxylated on a proline group, and when that happens, it becomes a target for the proteasome. So the lifetime under normal circumstances of this HIF-1 alpha is less than 5 minutes in tissue. And we looked at this in breast cancer, and we could actually visualize where this protein was and how much of it was in animals at different stages of onset of carcinogenesis. So it's a crucial enzyme. We were just delighted when people found that there was a mutation in it, and it helped to explain things. Even more interesting, the partial product that's going from isocitrate to alpha-ketoglutarate involves a step producing hydroxyglutarate. And it turns out one of the isomers of hydroxyglutarate is actually oncogenic. So yes, it's extremely useful. Metabolomics, extremely important. And now we, as I said towards the end of the talk, the really exciting part is being able to do imaging mass spectrometry of small molecules and large molecules. We started doing this at UAB in the context of what was happening in cataracts in the lens, and we were able to do imaging of the phospholipids and different levels of acylation of phospholipids, and we're able to do that spatially. So there is now a new work group in the Metabolomics Association of North America that's just been started that is pioneering discussions on imaging metabolomics. So if anyone is interested in that, I'd be happy to tell them about it. We meet once a month on Fridays, the second Friday of the month. So just send me an email and I'll be happy to tell you more.
Well, there was an earlier question about when you're talking about different samples, whether that was only blood samples or if there were other samples.
Oh, it can be anything. As I said, we were doing fecal samples. They proved to be really, really interesting. When I was first asked to do that by a guy who was here at UAB who was looking at children with spondyloarthritis, and so they had collected fecal samples and sent them to the microbiome facility, and they were sitting in 20% glycerol. And when I told my lab we would try to measure them, the lab was not happy. They said, impossible. Well, we found a way. We did an extraction to ethyl acetate, got rid of all the ethylene, all the glycerol, and got beautiful data, absolutely beautiful data. And we found out what was going on. It was about the bacterial metabolism and other metabolism of tryptophan. And so we did the study twice and actually got the same answer both times, and that was published. So it can be the biofluid or the tissue can be anything you want.
Well, it's good that rather than looking at specific analytes, you're looking at a whole profile, so you can see everything in there, even if it is a some solvent. Unfortunately, Jim Semino had to leave, but he said that he had.
Yeah, I saw him on there.
Very grateful for your talk. Yeah. So, any questions from anybody else? We're at time. I don't know if you have another minute or so, and I see that some people have had to leave to go to another meeting. But any final thoughts, comments, questions? If you can also get in touch with Dr. Barnes by email if you have any follow-up questions.
Yeah. So we're
Well,
working, yeah, so we're working with a lot of people here at UAB who have NIH grants and many who are trying to get NIH grants in metabolomics. So we have a very active presence, and then we also work with people outside of UAB, some of whom were here at UAB and went to other institutions and decided we were the best. So I'm really glad to be able to continue to work with them.
and we're very fortunate to have you be able to teach everybody about metabolomics and
Yeah.
collaborate.
So look out for these workshops in the fall. For those who want to get in and try and understand the data analysis part of it, be happy to see them there and encourage them.
Yep, thanks. And thank you very much for the talk and everybody who has stuck around to the bitter end. Yeah, everybody have a good weekend. I know it's a holiday weekend for some folks. Just a note that next week will be the last Power Talk of this semester. After that, we're going into finals week, and then we will have a break and go back to fall. But we have one more talk for fall. So, all right, thanks everyone. We'll see you next time. Bye-bye.
Okay, bye-bye.